1 o'clock, and you will find that I am extraordinarily precise about time. I will be up here lecturing at 1 o'clock. So we're going to start the class. My name is Dr. Choi, for those of you who don't know me, and you're in functional neuroanatomy, just in case you weren't certain. Your D2L is already loaded, complete for the whole semester. I have notes in single slide format and a double slide format. Those are exactly the same. Some people just like the bigger picture, some people don't. So I put them both on there for you. The syllabus is also on there. It's rather elaborate. So a couple of things I want to go over. I have recommended a Nolte textbook. Please don't buy it. It is way over the top. It is a great thing if you want to go to the library and look at it as a reference and read little sections of it. That's how I would recommend doing it, but don't buy it. It's just way more than you'll need in this class. But the other one, the photographs and diagrams Nolte, if you'd like to have that in hand, it's a picture book, so you'll kind of rely on it a lot in the Anatomy Wednesday lab. But students have found ways to get it online, so they'll bring their iPads and still have the same pictures. That's perfectly fine too. You'll just need something to refer to you, to your specimen. There's no lecture in the lab. It is sort of self-run, and then I'll have TAs and myself be running around trying to help you out, but otherwise you work in small groups, okay? Having said that, we have two TAs, Grace and Hunter, who will be joining us on Wednesday. They'll be your best friends for the first half of the course as we work through all the sort of pin and identification section of the lab. Having said that, lab is in 345, Schrader Complex. It is a BSL2 room, which means technically you're not supposed to bring in your water bottles and food. So definitely no food, because we have formaldehyde fixed sections that will come out. And then if you do bring your mugs and stuff, just stow them away. They're technically not supposed to be in there, but I won't tell. But I won't defend you either if you get caught. Okay? Note on your syllabus that there is 2 oral quizzes scheduled for the first 15 minutes of lab. And that means I will take a randomizer, pull your name out of the randomizer, and you will stand up in front of class and answer a few questions about the lectures you have in here. Now, I know that sounds terribly mean, and you're already stressed out. Don't be. There's no consequence other than a little public pride loss, but the goal is to get you to study along the way. If you treat neuroanatomy as the binge studying the night before stuff, you will fail. I guarantee it. This class will not lend itself for that. It'll get more and more overwhelming. Now, there is no cumulative final, but the final exam requires you to know the material cumulatively. So we are going to diagnose patients. That is your final exam. I will play the role of all 4 patients that you must diagnose. I will provide you symptoms only if you ask me for those symptoms. If you don't ask me, I won't tell you, and you will never know. So in order to get there, you have to remember everything from day 1 to the last day, okay? So I encourage you to study immediately after lecture. Clean up your notes, figure out what you didn't understand, what didn't come out clearly. Come back to me, and I will explain to the cows come home whatever you need to understand. I don't have office hours. What I have is an Outlook calendar. Please familiarize yourself with how to use an Outlook calendar. Look on your Outlook calendar where you and I have openings, and you just make an appointment and I'll accept it or not. And then you and I will meet however long and however often you want to meet, okay? So the 2 oral quizzes are a little public shaming and a little public motivation, okay? No stakes, nothing to be lost there. Overall, the class has 4 exams, straightforward. 4 30-point multiple choice exams and 2 practicals. The first practical is what you'd expect in an anatomy class, pins and identification. And then the second practical is me. I am the 4 patients, okay? If you have ODS accommodations, you can have them certainly for the multiple choice exams. Please schedule them with ODS. You will take them over at the ODS office. But the 2 practicals, it's not possible to do ODS, so those are practicals, not exams, okay? practical exam. It's something that worries you all. It's worth more points, and there's only one. We do the week before a practice practical so that you understand the serpentine arrangement and all of that. What I have done more recent in the recent years is allow students to count that as their exam score. So if you want to go for it early and be free of it a week or two early, you may then take the practice practical and say that this is my exam. You can do that after you see your score, okay? But once you determine that, you don't get to take the second one, the real practical, if you change your mind. Because if you do, then we're going to just take the practical. We're not going to let you choose between the two scores, okay? All right. Because we have a mixture of students, we have PhD students, master's students, and undergraduate students all together in this room, we have to have slightly different sort of assessments and things to be graded on. For those of my 2 PhD students sitting in the back of the room, you must give lectures. So you will come to me when you have decided on the lecture topic, and I will schedule you in at the end of the semester. Your lectures will be on the exam, so nobody gets a checkout. They will create multiple choice questions that I will stick in at the end of the final exam, okay? For the master's students who are in here, and there's quite a lot of you, 6, 7, maybe 8 of you, you are responsible for creating an infographic for me for the first 5 lectures. Okay, so we will sit down and I'll explain what that is, but it's a 1-page graphic on what you've learned in 1 lecture. Okay? Let's see, what else? Everybody has a chance at 5 extra credit points. I generally never give out extra credit, but I have been beaten into submission by many semesters of students. To get your 5 points, you must submit 5 really good Kahoot questions. Multiple choice, as you whatever the character limit of Kahoot. And we will have Kahoot game day and compete for prizes. If I don't like your Kahoot question, I won't include it and you won't get that point. So if you make a lousy, simple, what is that thing on my shoulder, then I won't give you a point for that question, okay? I think that's it. Oh, I will not be recording Teams of my lectures. You cannot attend my class through Teams unless you are deathly ill or for some reason I will listen to your explanation, but generally it's a no. So you are welcome to bring out your recording devices, your phones, whatever you want. I mean, I don't care, okay? I don't care about attendance. I only care about how well you do on the exams and whether you learn it. So however you learn it, somehow if you can do it remotely, I don't know how you do that, more power to you, okay? So I won't take attendance. I don't care if you're here. I only care that you get the knowledge, so get it however you need to get it. Again, I'm here to answer all your questions and clear up any fuzzy neuroanatomy, but I will not reteach it to you in my office, okay? So just keep that in mind. If you don't come with questions, I'm not gonna just help you out that way. All right, any questions for me as we get going? All right, I speak really, really fast and with great enthusiasm, I hope. All right, so we're gonna hit the ground running. This lecture is a very broad appreciation of the brain. It's all the little bits that don't fit well into another topic. So it'll be a little bit of here, a little bit of there, but it's to give you a little orientation of where you ought to be. So we're going to start with the biggest division of the nervous system, which is the peripheral nervous system versus the central nervous system. Now, the peripheral nervous system basically means it is a method of communication for the somatic part of your body. So soma means body, right? That's literally what it means. So the somatic means everything below your neck or your head. So it has a sensory input side of things, so sensory receptors and neurons, and it has then the opposite, the motor output. If you cannot read my handwriting, you must tell me right away. It is sloppy and I'm writing really fast, right? But I don't mind if you say I don't get what you're saying. Remember, motor output, these include axons, neuromuscular junctions of the motor neurons, etc. And then we have a third one. This is the autonomic component. And the autonomic component is specifically to very specific types of muscles: smooth muscle, cardiac muscle, so smooth and cardiac muscles, as well as glands, secretory glands, glands that make hormones or other types of signaling molecules. Now, the autonomic nervous system is basically divided into the sympathetic component, the sympathetic nervous system, and the parasympathetic. And we're going to dig into this in some degree of detail. So sympathetic nervous system, parasympathetic nervous system. There is a third, but we really won't, I'll mention it later, but it doesn't really have much role because it's extremely autonomous. There's no way to control it, and that's your gut. So sympathetics, you know, as the fight or flight, and the parasympathetic, we call it the rest and digest. So we're going to get into the anatomy. It makes a very large impact on neuroanatomical function, surprisingly. Okay? Now, the central nervous system is basically what's left over, which is your brain and spinal cord. Okay? So we have spinal cord, and then the brain. Okay? Now, the spinal cord, we know, has axons of sensory information coming from the body back up into the brain. But it also has cell bodies and axons for the motor system. So the motor pathway, yes, we have motor neurons in the brain, and those axons travel through our spinal cord. But the spinal cord, actually the majority of the composition of the spinal cord are interneurons, are motor interneurons. So there's a lot of cell bodies in your spinal cord, and we don't know if to give it credit for. So in that sense, we have the motor output either by axon or by cell body. Now, the brain, we're going to talk about the brain. What makes neuroanatomy really confusing is the way we treat the nomenclature of the brain. And it kind of sucks, but there's really no way around it. It's been established, and this is how it works. We can look at the brain from a developmental standpoint. Okay? And so these tend to be larger syllable words. And we intermix, and it's not clear why in some cases we choose a developmental term versus a histological term or a gross term, but it's best if we as a neuroanatomist learn all of them, okay? So there is the developmental term, there is the gross, which means visibly detectable, visually apparent, gross term, and then there is the histological. Now, we can do functional neuroanatomy from all three viewpoints, and they will sound entirely different. And the fact that we mix and match them is what makes neuroanatomy more complicated than it needs to be. Okay? So, let's look at each one separately. The developmental terms. When we were a single-celled organism and then became 2, 4, 8, 16, etc., we then become a neural tube. So that clump of cells eventually finds its way to group together in a way that creates a hollow tube, and this is what you start to look like. Now, this is where the developmental nomenclature comes in. We have three basic divisions of our neural tube: the prosencephalon, the mesencephalon, and the rhombencephalon. Okay? So we'll often still use this type of jargon, but not all of it, just some of it. So for example, the prosencephalon we divide into the telencephalon and the diencephalon. So you'll often see elements of the telencephalon, certainly the diencephalon. We use a lot of diencephalic. The diencephalon is our hypothalamus and thalamus, so you'll see that terminology come up. The rhombencephalon is divided into the mesencephalon and and the myelencephalon. Okay? And then the mesencephalon stays mesencephalon. Okay? But we'll see that come up. Mesocortical, those kinds of terminology will come up. But now you understand that they represent basically these three divisions. Now, the prosencephalon, the telencephalon, diencephalon are basically our cortex, everything that's above our brainstem. This is the telencephalon is the cortical area itself, and the diencephalon is all the subcortical area. The metencephalon and the myelencephalon are termed basically pons and medulla, so those are brainstem. And then the mesencephalon means middle, so it's the midbrain. So we'll get to know all of those from the gross anatomical side. But they have their names that kick in for whatever reason. It's just habit and current style. Okay. Now here's the gross. The gross anatomical is the names come from sort of how they look, where they're positioned. So again, this is our rhombencephalon here, but in the gross term we call them pons and medulla. We'll call them the brain stem. The midbrain is this small section here. That's our mesencephalon. Okay, so midbrain, mesencephalon, those don't change too much. And then everything else is the prosencephalon, or what we are now calling the cortex and the cerebellum and so forth. Okay, so just so you know, that's where they stem from. Lastly, the histological side was created by Corbinian Brodmann. He was a German anatomist at the turn of the 20th century. So the early 1900s, had a microscope and decided that he was going to look at the brain through his microscope and created maps. And so he made over 50 subdivisions based on whether the cells looked the same and when they started to look different. Were they large? Were they small? Were they spindled? Were they pyramidal? And he created these distinctive sections. What's crazy is that from the 1900s, this map is still used. The map and the delineations are still accurate, and they represent a lot of functional important areas. So for example, we're going to learn about a half a dozen Brodmann's areas. You're going to be very quick on saying Brodmann's areas 6 and 4, 3, 1 and 2, 17, 18, 19. You'll know exactly what they mean, okay? So they're important because they allow us to make more delineation than just calling this the occipital lobe. We have 17, 18, 19, which is our primary, secondary visual cortices, et cetera, et cetera. So that'll lend itself, but you're gonna have to mix and match these three different ways of approaching anatomy, okay? So you don't have to learn them all right now today. It's not for what the oral quiz is about. This is just giving you the lay of the land, okay? Okay, so occasionally I throw up this blue screen. It's a quick review. It should help you. If you understood what I just told you in this kind of encapsulated way, you are following along with me. If this doesn't make sense to you, this is a great place to ask questions. I'll go back. But if not, then if you want, you can call me aside and say, I don't get it. Oh, something I forgot to mention. Since I notice the classroom is empty before class, I'm going to come to class maybe 20 minutes early, and I'll be available here to answer questions. So if you'd like to come early and say, I got 1 or 2 questions from the last lecture, I'd be happy to spend that time with you, okay? So what we basically have gone over very quickly is that the neuroanatomy is based on developmental, gross, and cytoarchitectural or histological foundations in nomenclature, okay? Okay. All right. Now, let's talk about layers of protection because we don't often, there's no other place to put that topic. So the brain being such an essential organ that we really can't survive without, and we really don't want to bonk it too much. We have a number of layers of protection that have evolved along with us. So obviously the first one is our bony covering. So our brain is covered by a very thick set of bones, the skull, and then our spinal cord is encased in vertebral columns. So we have the skull and vertebral columns that encase our spinal cord in this kind of elaborate structure, and then the spinal cord is in this tiny little cavity inside. Then we have a couple of other layers of protection, and these go to our membrane covers. Okay, so in the membrane covers we have dura, arachnoid, and pia. So you'll see or you'll hear me refer to these layers as the swim cap, the saran wrap, and then the spray coating, okay? And you'll see why I call it that. But each of these three create layers of protection around your brain that serve different roles, okay? Then the last one we have in terms of protection is cerebral spinal fluid, CSF. Cerebral spinal fluid, remember you are a neural tube, so therefore you have space inside your brain. And so this canal system, this emptiness, needs to be filled with something, otherwise your brain would collapse on itself. So to avoid that, we have fluid inside the ventricular system, those cavities inside of our brain, and then there's the same fluid that is bathing our brain all the way around it. So essentially your brain is floating. Yes, we are tethered a little bit, but you are not resting, your brain is not resting on any structure. That would be bad if it were. And so this allows us to several benefits. One, it allows us to absorb pressure changes. So of course, if I bend over and I stand up, we sometimes see stars, there's a difference in pressure on my brain between those two states. Fun fact, the people who study this the most in terms of cardiovascular mechanics are when they study giraffes, because their heads change quite a lot of different distance when they bend over and stand back up. So the amount of pressure on their brain and the amount of work their heart has to do to pull it off is really amazing. It's like going off into space. Cerebral spinal fluid also allows you to do that crazy stuff people like at your age like to do. And when you go dancing, you ride on roller coasters, you're doing your skateboards. I see a skateboard here. Anything that jostles your head, you don't want your head or your brain to be slamming inside to the hard surfaces of our bony covering, so that fluid surrounding kind of creates a kind of bubble wrap. It allows a little bit of buffering and a little bit of cushion. So you've got a waterbed cushion around there. But you have to remember, our brain is in such a tightly regulated system that even your heartbeat, the thunk of your heartbeat, creates a pressure change in your brain that has a brief, for every beat, a brief increase in pressure and then a release of pressure. So we need the cerebral spinal fluid to be there to absorb even the small change created by a heartbeat. Okay? So 3 layers of protection. Questions? This is the pace I speak at. I know, it's very fast. Okay, good. All right. So let's look at the brain coverings a little more carefully. This is dura mater. Okay, and in Latin it translates to top mother. Okay, and top mother is tough. If you've ever noticed in the Gross Lab, if you've worked with brain tissue, this covering you really cannot tear by hand. You have to cut with a knife or scissors, but if you try pulling at it, it's not something that's going to easily work in your favor. It is a tough membrane that is mostly made of collagen. So it is a collagenous material that adheres firmly to the inside of your skull. So this is one of the major tethering components that keeps your brain from, for example, spinning around inside your head and pointing in the wrong direction. Your dura kind of keeps it oriented in basically in the correct place. Now, the dura is made up of 2 membranes, 2 dura. There is the periosteal side or periosteal membrane, and there is the meningeal membrane. So throughout, you have two pieces of dura, okay? They're mostly working together, okay? So in this diagram, you can see here, the blue is your dura mater. They're being exaggeratingly clear about the two layers. You don't normally see it like this in any way. You can see the two layers, and then occasionally the two layers will separate from themselves. But right now, the outer one, the one that's closest to the bone, is the periosteal layer. The one that's closest to your brain is called the meningeal layer. Okay? Now, other fun facts about your dura mater is that the periosteal membrane has most of the blood supply. So your dura does have a blood supply. It's there not to feed the brain, but basically to feed the bone. So the inside of the skull is being benefited from that blood supply. The dura mater provides mechanical strength. So as I mentioned before, it prevents your brain from turning around inside. It has its own blood supply, although it's not meant for the brain, it's meant for the skull. But it's something important for neurosurgeons to know that the dura does need to be perfused, and more importantly, it feels pain. Okay, so it can detect pain. So it is the only part of a brain surgery that requires anesthesia. So a simple local anesthetic is all you need to have brain surgery. Otherwise, the other membranes, the second layers of protection, the arachnoid and the pia, do not feel anything, and neither does your brain. So most brain surgeries, especially the very complex ones, are done in awake individuals because you kind of want to make sure you're not touching something you ought not to touch. And so all they do is have a little local lidocaine anesthetic, and then they cut the dura, which can be stitched back together because it has a blood supply, and it will heal. Okay? Okay, that's the first layer, the dura mater, collagenous, very tough, inside the skull, has two layers, mechanical strength, blood supply, and pain. Now, the next one is called the arachnoid layer. It is the second one. In my analogy, as you know, the dura was our swim cap, so you can imagine it's really thick, transparent. But here, this is the saran wrap layer. So the arachnoid layer is the middle membrane. It's very wispy, it's very spidery, which is why it's given that name arachnoid. Unlike the dura, it has no blood supply. Okay. It does not feel pain. And like saran wrap, it does not follow all the nooks and crannies. So it adheres to the surface of the brain, but it does not dive into the sulcus. So the peaks, the curves of a brain is a gyrus, or gyri plural, and then the valleys of the brain are called a sulcus, or sulci. So it does not drive into the sulcus and follow all the sort of vertical terrain changes. And so because of this, the arachnoid creates a space. Okay? So before I tell you about the space, let me tell you about the trabeculae. So the arachnoid sort of bridges the space between the dura and the last membrane layer, which is the pia. And the pia is only a one-cell thick layer of a membrane, so it's not a great protective layer. So that's why I call it the spray paint. So in between the arachnoid proper is something called the arachnoid trabeculae. And the arachnoid trabeculae, you can consider them as collagenous pillars. These trabeculae are there to kind of create a structure. So imagine if we kind of made an analogy, maybe my arachnoid trabeculae, you can imagine it as styrofoam peanuts, right? They're just sitting in a box in every little direction, but they provide cushion And so that arachnoid tuberculae creates a nice space between the dura and the pia, giving it a little bit of structure and keeping things kind of off, don't touchy the edges, that sort of thing. Okay? So remember, this is a collagenous pillar that's kind of mixed in there like peanuts, styrofoam peanuts, inside the arachnoid layer, connecting the arachnoid membrane to the pia membrane. Okay? Now, if we look at the idea of the saran wrap analogy, remember the arachnoid won't dive into the sulci, so then it creates a space called the subarachnoid space. So if, for example, we could squirt a little dye underneath the arachnoid membrane, the brain would look like this. So notice here, we have blue filling in all the sulci because they are trapped underneath the arachnoid layer. So if we have something going on above the arachnoid layer, but under the dura, like a subdural hematoma, then your imaging would look not like this. You would not see blood going into all the sulci. You would just see a very flat layer. So we'll get into it. So this is why it's important to know the 3 layers, because in imaging it, and that's what this class is all about. What does it mean to a clinician to take this class, okay, or to come from this class? So we're going to take it from a clinical type point of view throughout. So if you're a clinician looking at imaging and you see all the sulci, then you know you are looking at a subarachnoid bleed or a subarachnoid injury. That makes a very big difference if it's a subdural or an epidural, etc. Those mean different outcomes for the patient, okay? Now, also what it highlights is some of our cisterns. So pockets of subarachnoid space are given a separate term called a cistern. So subarachnoid cisterns. Cistern, just, as you know, it's a vessel. A big bottle is a cistern, a jug is a cistern. So what you see here, we have a number of cisterns. There's a chiasmatic cistern, a pontine cistern. This is just where the space holding the CSF is a little larger, so we gave it a name. Our biggest one is here at M. This is a cisterna magna. Okay, it's very large and fat. It is somewhat accessible. You can push somebody's head down and insert a needle and just get it into the cisterna magna. If I can't imagine why, but I think it's done for some reason. But you are very close to all the other goodies in the brain, so we generally don't like to play around in that area. Okay? So, but anyway, just so you know that there are these cisterns, but they're no different than the space that you see up at the top. These little thin areas, it's all subarachnoid space. Okay. Now, the relationship between these 2 membranes is something to point out. Remember, we have 2 layers of dura. Okay, this is the periosteal version, and this is the meningeal. And occasionally, our 2 areas will separate. Okay, and they will create its own sinus, a cavity, a space. So this is the central or superior sagittal sinus. It sits right on the top of your brain. It's a sort of tube-like cavity or cavern, and it's usually filled with venous blood, right? So this is old blood that's on its way back to your circulatory system to be rejuvenated by your heart and lungs. Now, underneath our meningeal dura, you'll see our arachnoid space, right? Here's our arachnoid membrane, and then sort of creating by our styrofoam peanuts, the arachnoid trabeculae, creating this kind of nice cushiony space. And then the pia is right hugging every nook and cranny of the brain. So that's my spray paint, right? It's on every nook and cranny, follows every shape. Now, this is full of cerebral spinal fluid. We must move the cerebral spinal fluid. Cerebral spinal fluid is newly made every minute, and therefore we have to get rid of it in equal pace. So how do we get it out? Well, remember, we can't, in order for cerebral spinal fluid to leave, we've got to go past the arachnoid, get past the big thick of dura, a layer of dura, and then we have to get past this endothelial lining inside of our venous sinus. So we got 3 membranes to get past. It's not possible. So evolutionarily, we created what's called arachnoid villi. Okay? So arachnoid villi are deliberately created channels that push through or basically are allowed through the dura. And these are one-way channels, so CSF can leave, but blood from the venous sinus cannot come back in. Okay? So this allows our circulatory system to be maintained. Okay? Questions? The last, as I've told you, is the pia mater. Tender, pia means tender, so this is tender mother. And tender mother just lines every shape of our surface of our brain. Doesn't do a whole lot in terms of protection, but it's there. Questions? Okay. So, quick review, protective covers, the bony cover, the coverings, the meningeal coverings, dura, arachnoid, pia, and then of course the cerebral spinal fluid, which we know is going through our ventricle system and that subarachnoid space. Why do we care? So this is why we care. So we can then create spaces that are not supposed to be there. For example, if you have an epidural hematoma, it is a space because of injury that's created between the bone and the periosteal layer of the dura. So that type of imaging is different than if you have a subarachnoid hematoma, which is between the dura and inside the subarachnoid space, underneath the subarachnoid, okay? So you could get imaging that looks like, if you see blood in this kind of shape, then we know that it's under the dura, okay? Because it's taking on the shape of the dura. If we have one that's a little more compressed, it's not flowing under the shape of my dura, then it's just above the dura and it's only pushing in a more round shape rather than it's more long, oblong shape, because the oblong shape can then follow the contours of your dura. A subarachnoid hematoma, or you all know that hematoma means like the blood, collection of blood. You can then start to see the sort of patterns of the cell side so that you know you're underneath the arachnoid layer now filtering in all the cell side. And then of course there's within the tissue like a DA, an axonal injury. Okay, now because the dura is a two-layer membrane, and we know that it can open up between the two. I have the periosteal, and then underneath I have the meningeal, right? And I can create a sinus in the middle. Another thing that this can create is a reflection, and that's when the inner meningeal layer fuses together and then creates this sort of divider, okay? So our biggest dural reflection, that's what they're called. So it's a dural reflection when two parts of the dura fuse together. This one is our biggest one. This is the falx cerebri. Falx means sickle. So that's that ominous weapon held by the Grim Reaper, right? The sort of farming tool, the scythe. So this is a sickle-shaped dural reflection. And I'll show you a nicer picture, next picture here. The falx cerebri is anything in blue. So basically what it does is it is dividing the two hemispheres. So the falx cerebri sits in here like this and creates a divider for our two left and right hemispheres. So our hemispheres never touch each other at any point because of the falx cerebri, right? So our dura is otherwise covering on top. The other important one to remember is the cerebellar tentorium cerebelli, right? So the falx cerebri splits and becomes a Y, and you can see the tentorium cerebelli in green. You can see this in the actual picture. So once it starts to curve here, this is the left side, and then you can kind of see the one inside that kind of split. And their job is to separate the parietal temporal lobe here from the cerebellum. So my model doesn't allow me to stick a piece of paper in there, but we could put the cerebelli or tentorium cerebelli here and here. So you can see this would split and become two reflections. The other important reflection that we want to know about is the diaphragm ocelli. The diaphragm ocelli is not labeled on here, but it is right here. Okay, diaphragm ocelli is a skirt that covers the bottom ventral portion of your brain. So here is your pituitary gland. You know that it sticks out and it's not really part of your brain. So we have a membrane that covers everything but the pituitary gland. And it allows this little hole, these are the two optic nerves, this is your pituitary stalk. It allows the pituitary stalk to go past, but otherwise, when you look underneath the brain, you'd see nothing but a pituitary gland. It should be covered up by dura mater and this dural reflection, okay? So there are three I want you to know: the falx cerebri, the tentorium cerebelli, and the diaphragm ocelli as three examples of dural reflections. Okay. Next, we're going to talk about the blood-brain barrier. Okay? We know we say this a lot, but a lot of people don't understand what the blood-brain barrier is. It is not an actual barrier, it is a conceptual barrier. It's created by three different ways of blocking something from getting to your brain. So the blood-brain barrier was discovered long ago when they were experimenting with the cardiovascular system in animals, and they would inject somewhere in the body like a heavy blue-green dye and look to see where it went, to just kind of track your circulatory system, where your arteries and veins were. But they also noticed that no matter what they stuck in that system, the brain never got colored. So they realized that the brain has its own special barrier, that it could not accept a lot of stuff that was coming in through the circulatory system, which was good. So we have how to appreciate the blood-brain barrier and appreciate how we can stop things that are in the circulation system to get in. So we have to consider, how does blood infiltrate our brain anyway? So there is the extracerebral capillary route, there is the intracerebral capillary route, and then there is the choroidal capillary route. So these are three types of capillaries, blood vessels, that are nice and small, that can have access or come close to having access to our brain. Extracerebral means it's outside of the brain, but it's sitting on top. So maybe it's in the dura, or it's a big blood vessel that is just sticking to the surface of our brain. And you will see that a lot in the brain specimens. You'll see big blue vessels that are covering our brain. So our brain needs a lot of blood products, oxygen and glucose, but we don't like it to get too close. So we have extracerebral capillaries. Now, when something comes out of that capillary, other than what the brain wants, glucose and oxygen, it has a way of blocking it via the arachnoid. So the arachnoid layer becomes the first line of defense for anything coming out of the extracerebral capillary. So this is a brain. I know it's very unusual looking brain, but it's just sort of a schematic of a brain. So the reason is the extracerebral capillaries, they are fenestrated, meaning they have these little apertures, these little windows, pockets that allow stuff to come out. So since we cannot prevent this from happening, the brain has evolved to have the arachnoid serve as a barrier. So even if something came out of these apertures or these fenestrations, it's going to be met with the arachnoid. So that's our first blood-brain barrier. Now, for capillaries that are already embedded in brain tissue, so these are intracerebral capillaries, we're already past the arachnoid layer. So instead, these vessels have evolved to already have tight junctions. So tight junctions inside intracerebral capillaries is the way to prevent stuff from coming out. So in this case, there are no little holes or apertures or fenestrations that are available to them, so they stay inside the capillary. The last is the choroidal. Choroid is important for the synthesis of cerebrospinal fluid. So we have to have blood, okay, because that's where we extrude and create CSF. So we have these choroidal capillaries, and then we have this layer of pia that stinks as a membrane, so everything gets past the pia. And so what becomes the membrane barrier is the choroid epithelium. So the choroid epithelium is the barrier for anything pushing out of a choroidal artery. You can see here the choroidal arteries or capillaries are also leaky, have little apertures, but when something comes out, they get blocked by the choroidal epithelium, okay? So the blood-brain barrier is a functional barrier. Depending on what blood source, you have a different way of blocking entry. Either it's the arachnoid layer, either it's the tight junctions built into intracerebral arteries, or it's the choroidal epithelial, the lining of the ventricle, that creates a barrier for those vessels inside that part of the brain. Questions? Okay, now, oddly enough, we've made a great blood-brain barrier, and then the brain has deliberately created circumventricular organs. This is where there is little to no blood-brain barrier. So, I know this is a list. All you have to do is recognize this list on a test, okay? You don't have to memorize it like to be able to write it out yourself, but recognize it as a CVO, a circumventricular organ. So in red are the locations of the circumventricular organs where there is very little or no blood-brain barrier, and the blood vessels in these areas are allowed to dump stuff out and be kind of interactive with the brain, or vice versa. It's mostly body to brain. And it's sort of useful because where they're positioned are areas that pick up stuff that produce hormones. So you kind of want to know what's going on in the body, sample the blood, say, oh, we're having that experience, let me create a reaction to it. So these circumventricular organs, like the median eminence of the hypothalamus is a big producer of lots of stuff. The pituitary gland, again, produces lots of stuff. We want to be able to sample what's going on in the body in real time and react to it, okay? And so we have areas in the brain surrounding these CVOs that are highly reactive in terms of producing a secondary or reactionary response, okay? So just learn the list. There's nothing more exciting than that. Okay, so here's our quick review. The blood-brain barrier, not a single barrier. It's a functional barrier. Depending on these three entry points, you have different ways of stopping something. And then, of course, we do the opposite. We create holes in our system, circumventricular organs, that deliberately allow stuff into the brain, at least to be purposeful. So this is, this is nice against microorganisms like germs and viruses, but it's not good for antibiotics or chemotherapy. This becomes a problem for treating brain tumors and injury. Okay? All right, really quick. We don't do neuroscience in here. We do neuroanatomy, but I have to show it. Here's a neuron. It's a body. It's an axon. It's got lots of dendrites. That's all you get. Okay? Other cells that are not neurons in your brain are called glial cells. So these are all glia, and they're all different kinds. They're the astrocyte, oligodendrocytes, and microglia. Oligodendrocytes produce and make, or not produce, they become myelin. They wrap themselves around neurons to myelinate them. So that's all you get. Okay, outside of the central nervous system, oligodendrocytes are not called oligodendrocytes, they're called Schwann cells. They're just the peripheral version of a myelinating astrocyte or glial cell. And then this will become important later. You gotta know your orientation. Dorsal versus ventral. Dorsal fin, ventral fin on a fish. Rostral, caudal, okay? Superior, inferior, anterior, posterior, We're gonna use these interchangeably. At no point will I define them again. Just ask me when you can't remember, okay? Last slide. Gotta know your planes, okay? A horizontal plane is horizontal. A coronal plane is like a corona. You know those Byzantine arts and people's halos were these circles? So this is a corona. Coronal direction, horizontal direction, sagittal section, parasagittal. Okay? So you need to get to know those, and then I'll teach you the last few words. Bye-bye. See you Wednesday, 3:45 in lab.